So I want to start with the idea of innovation. When you think about medical innovation, sometimes to me, and you're to me a new generation of innovators, and what you're doing is very innovative. Sometimes it feels very incremental to me, like, okay, we're making innovation, but we're building on the last study, and it's little by little by little. You know, I come from the tech world where the disruption comes from the bottom, from this underserved, unloved customer, and then, you know, you can be a Snapchat over a Facebook or something like that because you're serving a whole different community or you're looking at it differently. So, what are the factors of innovation in myeloma? And how do you develop more disruption? Because you are, to me, you are.
You want to go first, Dr. Langren? Hola.
No, why don't you go first?
So I think that, this is a very good idea and a very good thought. And I think that the limiting factor here, why it's so different, why this is so different than tech, it's because there's the issue of safety. And so what happens is that we said we're not gonna hurt people. So we have to be careful when we do our innovation that we're not hurting people. And so what has happened, for example, in the smoldering world and in myeloma precursor disease is that investigators and physicians, they don't want to hurt people. And so for a long time, physicians were saying, well, what if you treat these patients and then they do worse, right? And nobody knows. And so there's a whole discussion about that. It doesn't look like that's the case, okay? It looks like actually you treat them with these very effective medicines, they tend to do better, actually. That's what it's turning out to be. But before we can do that, we have to, build data. And so what happens is when you try to do a clinical trial, this clinical trial is regulated, and it's regulated in terms of the institutional review board. So there's an ethics committee that decides when this trial is going to come out and how, and and they go through everything and they ask their questions, and there's a scientific review, and there's the FDA that also regulates all the safety. So there's this whole thing built around clinical trials, which is how we innovate in medicine, that is difficult to basically bystand, to go through without it, because in the past, investigators and physicians have done clinical trials without going through these venues, and it has hurt people. And so I think that we could think about how to move through these venues a little bit faster. That would be great. But all these things that are in place for patient safety are very important. And the key is, how do we implement trials? How do we do this? I have a trial that it's taken me 3 years to open. It just opened. It's taken me 3 years, right? So we thought about this trial in 2018 or 2019. It just opened. So, and strategies to do that. Dr. Langren, what is the solution? Tell me. Because
does this microphone work here? So I think that's a very good question you are asking, Jenny. Why is it not like tech? And I think Elizabeth, of course, puts her finger on the first thing that we were told in medical school, which is critical, is do no harm. But I also do think that there is a lot of opportunity beyond what's going on. And I think my answer to your question there, Jenny, to build on what Elizabeth said, I agree with everything you say, do no harm. There are a lot of factors always have to be taken into account when it comes to development of the clinical trials and so forth. But I think the new technologies that we have to investigate disease are dramatically changing. I've been in the field for many years. I have had many ideas for a long time that I could never test. So to be very practical, to give like some simple examples. So for example, I'm fascinated with the genomic characterization of the disease. You can do whole genome sequencing or single cell sequencing, but the technologies that we had, say 12 months ago, or 18 months ago, they would require a certain amount of DNA in order to run. So to be practical, you needed, say, 200,000 cells or more to do the test. I wanted to investigate the monoclonal gammopathy and the smoldering myeloma. If you did a sample, you ended up with 10,000 cells. So in the patient sample where you wanted to ask the question, you couldn't do the test. That has not changed. There are low input DNA technology. You can do it with a few hundred cells. You can actually ask the questions today. We couldn't do it just a year or year and a half ago. So I would say all these new technologies, they are here now and they're changing the field. I read papers online that are in the preprint servers. You can do whole genome sequencing in 24 hours for $100. It's amazing. It's just changing everything. Now, what's going to happen is that we're going to generate so much data. So if you do whole genome sequencing for 3 patients, that's 1 terabyte. And I think a laptop is half a terabyte. So for 1.5 patients, the hard disk is filled. And no computer could open a 1 terabyte file or half a terabyte file. So the computers we have don't work. So when I'm ordering laptops to my team, their laptops cost $10,000. We don't order from Best Buy or Amazon. They have these super expensive. We need to have that. You also need to have the right tools. And you need to have the people that understand. So I have recruited people from Sanger Institute in the UK, from Sloan Kettering, and from Boston, from the broad. So I think technology you need to have the critical mass of the smart people that understand how to analyze it. And we also need support because it's very expensive to do these things. So we are in our environment supported by the university, and we are constantly applying for grants and things like that. So I think my answer back to you, Jenny, is technology and new medicines, critical mass. You need to have a team that is focused and thinks about the same thing every day and pushes the envelope. And then you need to be in an environment where there is support to go forward. So things don't change. The support doesn't go away in 6 months. Someone needs to believe in us. And here we have the support from the institution, and you have support from your institution. So I think those, to me, are the critical factors. And I think we will see earlier detection of disease through new technology. I think smoldering myeloma is probably going to go away. I think we'll have monoclonal gammopathy in myeloma. I think we'll have MRD driving treatment decisions, I think. I know this is controversial, but when I heard Nina talking about cell therapy and immunotherapy, I think a lot of the transplants are probably also not going to stay the way they have. Let's see what the other think. They may not agree with that, but I think that a lot of change will happen. I think a lot of the drugs will probably be non-chemo based. I think we will have chemotherapy-free treatments for myeloma. All these things are happening right now. It's driven by technology. It's driven by very disruptive science. That's my take.
I agree with almost pretty much everything except the transplant. No, everything. No, one, you have to take risk. So if you know of a trial, and a lot of you have taken the risk to be on a trial, you have to take the risk, and we have to take the risk, and drug companies have to take the risk to invest in these technologies. Two, Elizabeth hit it on the head. It takes forever, A, to start a trial, and B, to conduct a trial. And in myeloma, we're doing great, C, to get the endpoints of the trial. So I agree with Ola, we need to do endpoint management better. We have to ask the FDA, hey, it's okay to use sustained MRD negativity, which I think is a great surrogate for how people are going to do to get answers quicker. Because for everybody who's in this room, all the answers that we have are outdated now, if you can believe that. Because now, for example, DARA-VRD is better than VRD by a randomized trial. And that's a phase 2 randomized trial. And and a lot of us would rather use DARA-KRD, so there's just, we're moving faster than the trials are letting us do. I am a huge proponent, and I don't know if patients and advocacy groups can help us. Clinical trial machinery has become worse than the federal government. It's so difficult to get things done, approved, changed. I know safety is a huge thing, do no harm, but also do no harm by withholding drugs that are effective for patients. So if you withhold efficacious drugs, then you're doing harm, right? So we shouldn't have a pathway that does that for extended period of time. So I hope that we can all work together to trim the fat, really, of clinical trial management, because I think that's one of the things that's really we could do better holding us back. We're all Elizabeth and I, we're millennials, as I say, and we're impatient. So we're not going to stand for this, I don't think. And I think we'll probably get really impatient and want to use all these new techniques that Ola's talking about to get answers more quickly. So if anybody has the possibility of participating, for example, in a trial that looks at bone marrows versus blood for detecting MRD, that would be a great trial to participate in. And that's not even an interventional one. So we could develop a new blood tool to get answers quicker. So that would be another way that we could all work together to get answers quicker, move the field quicker.
Yeah, I think you look at clinical trial design, maybe, I mean, in the myeloma space, you've even had drugs approved after phase 2. So that idea of getting early approvals would move things faster. I think even changing inclusion criteria or looking at like, I don't know, I think younger people have a tendency to kind of go, why are we doing it this way? And can we do this differently, you know? And is this the right way of doing it? And so I just appreciate all that. I think I want to follow up on what you were talking about MRD. So can we have a conversation about that? Using MRD as a new clinical trial endpoint and what that means to patients, so patients are aware.
Yeah, so I do think there's been a lot of talk about MRD. I worked on it for over 15 years. If you attend a meeting these days, if you don't hear MRD being said every 10 minutes, you're not paying attention. Everyone is talking about it all the time. I used to be invited to conferences 10, 15 years ago when we had debates, and I was always assigned to argue that it was important, and I lost every time. I went every year to lymphoma myeloma in New York. I always lost every debate, and I did so many of them. And there was a tipping point a few years ago when I started winning, and now I never get invited. They don't even have the debates because there's nothing to debate. Everyone says you must do MRD, and I've seen this happening for many years. So MRD is very important in myeloma. There are multiple meta-analyses showing that MRD negativity is associated with longer progression-free survival and also overall survival. And it's probably not rocket science, because what MRD is that you cannot find the disease. So if there is no disease, it's probably more likely to stay away longer. It's kind of makes perfect sense. So I think where the debates are starting to come back now, so I get new invitations to talk. There are a lot of other debates, so it's not that we never get invited, but the topics change. So what's happening now is that people are inviting us to talk about, could MRD drive decision-making? And I always get invited to argue for that. And I have lost a lot, but I started to win more and more, which I take as a sign that that's probably the way the field is going to go. So can you use MRD to step down the therapy? You treat a patient and the patient becomes MRD negative. Could the intensity of the treatment go down? I think the answer is yes. And it's not rocket science to me either. If there's no disease to treat, why do you go so hard on it? If the patient turn MRD positive again, could that be a trigger to restart therapy? And I think the answer probably will be yes. Are there 1,000 trials that show this? No, but it takes time to build that up. That's, I think, is an area where patients will benefit from it. But also MRD will be critically important for drug development. And I worked for all these 15 years with the FDA, and I work with them closely to generate the data that will give them the confidence to say, you open a clinical trial, you randomize to say 2 arms, you compare a new therapy with an older therapy, and you capture progression-free survival. That means patients free from progression. And if the new drug has a better progression-free survival, that will get approval. That's how the current landscape is for new drugs to be approved. Because the drugs are so good, it could take 4 or 5 years to generate all the data and to prove that this drug is much better. This is what happens right now. So it could be a great, great drug. And you give it to a few patients on a trial and you see, wow, this is so good. Well, you still have to do the exercise and feed the FDA with the data. It could take 6 years. We are talking a trial for relapse disease. You need to generate all the data as a company, and then the company needs to give it to the FDA and they will look at it. And these are the rules. So what we are trying to do is to say, if you could look at MRD status, if there are more patients MRD negative on one arm versus the other, and you have that information after 1 year, if that is associated with progression-free survival, you could make the decision after 1 year. So in this case, you would save 4 or 5 or whatever number of years. So think about it, all the new drugs that you have to wait 5, 6, 7 years for them to be approved, you could wait only 1 year. This has to happen. There is no other alternative, and the FDA knows it. This is very, very important. And I think we need patient support for the effort we have been working on for a long time to make this happen. This is critical for the field to go forward.
I want to just say about the MRD. I completely 100%, 1000% agree with what you said. And I want to make sure that we are clear to all the patients, or I'll speak for myself. MRD is super important, I think, to understand the depth of response and maybe to predict what's going to happen. What we don't know yet outside of clinical trials is if you make decisions on it, what happens at that time. And I just want to emphasize this because a lot of patients come to us with a lot of confusion about what's going to, why didn't you check the MRD? And I said, well, you're in stringent complete response and you have been already. And some patients want to get a serial bone marrow biopsy. Some people don't want to get a serial bone marrow biopsy. And it's complicated because we don't know yet whether we should make decisions based on rising MRD and what that threshold is, right? We have these criteria that we have from long time standing IMWG, International Myeloma Working Group. We use those criteria. I hope that my colleagues will use MRD to make a decision and have some criteria, but we also don't want to see that you can't be eligible for a clinical trial because you weren't quote unquote refractory to something or et cetera. So I don't want to confuse any patients to say that you have to get an MRD test or not, but we do as a field think it's important to move the field forward. And so it's just important to make sure you have this discussion because I'm happy to order a bone marrow on anybody, honestly, but usually the patients don't want to get one.
Right. Yeah, that happens. Very important points. And yes, of course, minimal residual disease is very important. We have many studies. We know it predicts outcomes. And, it predicts outcomes even in earlier disease. So the goal is, to have the sustained MRD negativity in smoldering myeloma. The FDA, of course, knows, and I think is very willing to do trials where MRDs are endpoints, and also to do those trials where you stop treatment if you're negative, or you continue or intensify treatment if you're MRD positive, if you still have disease. And they also have the accelerated approval program, which they have approved, many drugs over the past few years without what we would call like the hardcore endpoints, which are progression-free survival or overall survival. And this has led to early access of many drugs to patients. So we do, at MD Anderson, any newly diagnosed myeloma patient that comes to us goes on trial. This case is, we have a trial and Dr. Langren. We're comparing KRD to KRD data. It's a newly diagnosed study. It's open here also. And all the patients, we do also single cell sequencing. So we take a sample and we sequence those, each cell of those patients in the bone marrow for the myeloma cells and all the other cells that are there. And so those are, some of the things that Dr. Langren was showing here, how to do innovation coupled with how to, with MRD. So for these patients, for example, it will be very easy to see, and we will see this, is the patients that are positive versus the patients that are negative by single cell sequencing. So I think you showed some data already that you have for that. So, I'll be able to maybe give you some, or we can, So, that's what we're working on. But again, how to get all this technology and transfer it in a way that it helps patients, with all the regulatory background, to me is still challenging,
Okay, we have time for one more question. Is that okay? Sorry, Greg's giving me the hook. So you talked a lot about CAR T therapy, and now we have an antibody drug conjugate, bispecific antibodies, and CAR T therapy all going after this BCMA target. So with availability like it is, when it's really hard to either get into a CAR T trial or get the therapy itself, how do you suggest your patients navigate that? Like, I was just listening to another myeloma workshop at the IMS workshop that was talking about maybe using bispecifics. Maybe it's best to use bispecifics first and then do CAR T, or like, it's so confusing, right?
I have a lot of opinions on this. I'll keep it short. One, I think if I had everything all the same, I personally would try to get to CAR T only because you want to get the T cells as soon as you can. Okay, that's ideal, right? It's not going to happen for all of us. That being said, I do think that there's a role for using CAR T and bispecifics sequentially if you have to. We know that the people who progress with after BCMA CAR T still have BCMA on their cells. So it's not wrong to get a CAR T cell therapy and then get BCMA or bispecific T cell engager. And something huge that science that like Ola does, all these things looking at, maybe we'll find other targets. So things like GPRC5D, FCRH5, these are new myeloma targets that we can use maybe after CAR T or for a different CAR T. So super, super important to give samples because we really want to find these new targets that we can develop these effective effector mechanisms.
Thank you, Nina. I mean, I would agree that if you're, if you're a patient that is eligible for a CAR T cell therapy, that's what you should do because the efficacy of CAR T just seems better overall compared to the bispecific. So those are antibodies or to the antibody drug conjugate. So if you're a patient and you have relapsed myeloma and you can do a CAR T on a trial or outside a trial, that's probably what you should do. Now, as we all know, the spots for CAR T are far and in between outside of trials. And for the trial, you have to qualify. So if you're someone that doesn't qualify for a trial for CAR T and you don't qualify or you don't have a spot for a CAR T outside of clinical trials and your disease is progressing, then yes, of course, then, the next best thing would probably be a bispecific. So you would then look for a trial for a bispecific, or you would, actually, yeah, it's just a trial because they're not approved yet. So you would look for a trial for a bispecific. That doesn't work, then you would go to the approved therapy. The approved therapy is the antibody drug conjugate, belantamab, mafodotin, or Blenrep, and that's what you would do. And you can effectively do, if you're waiting for your CAR T spot or something, you could do the antibody drug conjugate. So you can do Blenrep and follow that with, CAR T. That could happen. We've actually done that and before transplant and things like that when we don't have any other options. And so there's a lot of things that you can do, but in terms of efficacy, the CAR T usually is the main one, then the BiTEs, and then belantamab. And this depends on the person and toxicities and things like that.
I think we all share a lot of perspectives on it, but we also have a little bit of a difference in perspective. So maybe I add some of my kind of difference in perspective. I think that, first of all, there is no data to prove that you should always do CAR T cell before antibodies, bispecifics. There is no data to prove that bispecific should always be done before CAR T cell. So it's an area of opinion. For that reason, I think in my practice, in my clinic, I always involve my patients in decision making, and I want to make them informed so they know what the options are, so they can make decisions that fit what they want to accomplish. So CAR T cells, you could argue and say it's great to have a one-time treatment. Unfortunately, for myeloma, we do not yet have curative CAR T cells. So it's not one and done. It's one, and then there is a longer effect. I think a lot of patients who've been treated with many lines of therapy are sometimes looking for a long time without having to be treated, but you still have to be tested. So it's not that you never come, but it's great to not having to come in for treatment for sure. I can see that. On the other hand, you have to organize, so there has to be a slot available. Right now, that is a big problem. I have a lot of patients, I have patients from other countries. They call me every week. They want to come here and be treated. We unfortunately cannot offer all these individuals coming. I wish we could, but there's shortage, not due to us, due to the manufacturing. So that's a reality. When I was at Sloan Kettering, we had 10,000 myeloma visits in 1 year. And during several years, we treated probably less than 100 patients for CAR T cell. So already there, this was not the bulk of therapy we could offer. So there's a huge restriction in availability. So if a patient has a slot, that's great. The patient will have to come and collect the cells. It takes 1 month to make the cells, and they have to then be given. So if all these things line up and that's the right thing, I think it's a great option. The antibodies are off the shelf. You can have a clinic visit today, and we can start therapy next week or later this week. We could start on Friday. and give a shot once a week. There is no wait. You just make a decision and go. So I think different individuals will pick different things. And I agree with what Nina said, that this discussion on which to use first is a little bit biased by the fact that the current versions go after the same target, the BCMA. But there is another antibody that is very far along, probably will be approved in 12 months, going after the target GP or C5D, with a long name. There is a trial that was just completed at Sloan Kettering showing that patients who were treated with CAR T cells with BCMA, 71% of them had a great response to a CAR T cell for GP or C5D. So if you have more than one target for both antibodies and CAR T cell, I think you could think of going back and forth between the different. We just opened a tri-specific antibody trial here. So I think there will be tri-specifics, there will be antibodies with bispecific partnered with co-stimulatory antibodies. So there will be multiple antibodies. There are coming many trials opening up with multiple antibodies. So it's impossible to predict what's going to happen. But one thing we know for sure is that it's just going to get better and better. There's going to be many immunotherapies coming. There will be multiple target CAR T cells with both GPRC5D and BCMA. So we have just started the journey here.
Yeah, super exciting. Thank you so much. And I'll turn it over to Greg.
So, here they are. Is this mine? Yes. I always put my cheap reading glasses down and I never remember where I put them. So this is really what I think is the best part of our program in many ways. So think about the questions that you'd like to ask. This is your chance. And we also will do this again this afternoon. And the way I'd like, it's like when you go to an auction and you sort of give them a little signal, just do that as it goes along and I'll let you know when you're in line. And we'll try to get as many questions in as we can. So with that, who would like to start? Oh, come on. Oh, there we go.
Thank you all for very nice presentations. I just want to kind of pick your brain on your vision. When I was doing my postdoc many years ago, cell therapy was a dream. Steven Rosenberg at NCI was dreaming about it, talking about it, and now cell therapy is here. So what do you think we are going to see in the future?
All right, I'm back. I'll just take this one. I think for the future, it's going to be quicker cell therapy. I think it's going to be more accessible therapies. If we're just talking about cell therapy, I think there's these point of care potential where you could get the patient's cells and then make them in vivo. I think there's just so many ways you could innovate cell therapy to make it more accessible, allogeneic, et cetera, and a better way to get it more accessible for underrepresented communities as well in clinical trials.
You want me to answer as well? All right. We said we would do one each, but it's all right. So I think that, of course, everything she said, but I think that we will have a lot more personalized treatment because the power of the technologies that he was talking about is amazing. The power of the single cell sequencing to analyze what the tumor and the microenvironment look like at the time that you are seeing a patient. I have never seen anything like it. And I think that when those technologies are more accessible, we're going to be able to give different treatments based on that. Definitely very potentially for relapse patients. I'm not sure how long it will take to move, but the technologies for analyzing the genome, the RNA, all that is just, it blows my mind. Every time I look at results, I'm just like, oh, wow, this is amazing. So.
Question back here.
I also want to thank all of you. I'm a patient myself. I want to thank Dr. Langren, who's my doctor, but I have a lot of questions from my family, my siblings, my children. It sounds like I have an early onset expert in front of me now. And what should I tell them? What testing should they do? When should they do it? I have nieces, nephews. They all want to know if they're going to get sick.
So Dr. Langren is a big expert on this as well. But I'm going to say that our recommendation, we usually do recommend to test. There's a clinical trial right now ongoing in the US where actually your family members can go in, register. It's a clinical trial out of Dana-Farber, and they actually send you a kit and they get tested for free. And that's one way to do it, which is a way for us to investigators to gather data. Or you could just have, your family members go and have the test done locally and then go from there. But if you go online, there's a study right now going on in the US where, again, you can have just everything shipped to you. And you just have to say that you have family members that have myeloma and you qualify. So that's something that they probably should go ahead and do. There are some limitations on the age, so you have to be above a certain age because we know that the incidence of myeloma and precursor disease, patients that are less than 40, 50 years of age is not much. So I would say that for them to check this out, to get tested, or go to Dr. Langren's clinic, get tested, go to primary care doctor, get tested. But I would say, yeah, I do say to my patients that if they have family members they're worried about, to just have them tested.
So what is currently known from the existing literature? So we talk a lot about genetics or genomics in myeloma. So there are two ways of looking at genomics for any kind of disease situation. One is to look at the disease cells. We also have all the other healthy cells in our bodies. That's the germline DNA. So if you think that the disease is caused by genetics, myeloma is not one of them. There is no evidence that myeloma is a genetically initiated disease. So that means that if one family member has myeloma, there is no evidence from the scientific literature that other people are genomically at risk in that family. There is not. Could that still be the case, and future research could identify? Everything is, of course, always possible, but people have looked and looked and looked, and no one has found any evidence. So the consensus in the genomic literature is that it's a so-called complex disease. Most cancers are complex diseases. Lung cancer is a disease can be triggered by smoking, but most other cancers are they're complex diseases. So that means that no one knows why they really happen. There are some risk factors that we have found in the past. So we did a study years ago. I was the lead investigator for a study investigating servicemen that served in Vietnam who did Agent Orange spraying. And we found that there was a higher risk of developing myeloma precursor disease after exposure to Agent Orange. Now, most people who have myeloma, of course, didn't do service in Vietnam. So that's not the only explanation. There are also studies showing pesticide exposure could play a role and things like that. So I think in families, we share both genetics and we also share environment because we live together. We also share behaviors. Families tend to do the same things. And no one really knows if there could be risk factors that are involving any of these. But I think importantly, there is no evidence genomically that people are at risk. And the last thing I say is that there are studies from Europe using very large databases with millions and millions of people where families have been created as kind of pedigrees in databases, showing that if a person has MGUS, there's another family member who has myeloma, that MGUS patient does not have a higher risk of progressing to myeloma compared to an MGUS patient who do not have a myeloma patient in the family. And that's a question that many people ask. So there is really no scientific evidence for genomic correlations at the current time.
Steve's next, and then if anybody else, signal you're next after that. One of the things that's interesting about these meetings that I've learned over time is this is basically a one-room schoolhouse. And in a one-room schoolhouse, you have novices and you have people who are very experienced. So one of the things, because some of you get everything and others of you are kind of like, what in the world are they talking about? And the point I want to get across is this is not to make you an expert. This is a process for you to start repeating and repeating and repeating learning about this. And then you're going to learn it. But the most important thing you should come out of this is not that you know a lot about myeloma, it's that you know what you need to do to ask better questions. Okay, that's the goal for today. So with that, I'm going to go to Stephen real quick.
Thank you, doctors. First question, real simple. Dr. Elizabeth, what is the proper pronunciation of your last name?
Manasang.
Thank you. I've heard people pronounce it different ways, and I like to pronounce it properly. On the topic of CAR T, I lost a very good friend because she couldn't get to the approved slot in time. I read that viral vector shortage is a problem. I read that the 2 to 4 weeks to re-engineer the cells is a problem. FDA certification of facilities is a problem, and manufacturing capacity is a problem. At ASH, there was a list of CAR Ts that were in the loop, and one of them was GCO12F, which they claim would have a 1 to 2 day manufacturing time. Is something like that going to make a huge difference?
Well, it would, except that it won't address the other 6 factors necessarily. Although I will say that even an approved CAR T that one of the CAR Ts that is approved, they're actually looking at a shorter manufacturing time to increase the number of slots, because if the production is sliced, then the number of machines that make these would double, right? So that's a mathematical equation. So all of the things that you highlighted are major issues. And what that means is that we're working on a version 1.0 of assembly line for CAR T cell therapies, right? There shouldn't be this many factors that are prohibiting the development of an effective therapy. So we need to work on a more efficient assembly line. And whether that means it has to be from a healthy donor, so we don't have to have the patients be the donor, and there will be better at allogeneic off-the-shelf CAR Ts, or whether that means that the production from the patient has to be much quicker versus this, like, Amazon send off and come back that takes up many weeks. Or we have to figure out a way for patients to maybe do in vivo generation. This is what I was talking about for novel engineering techniques. That could we give the patient the viral vector and their own cells know how to produce it in their own body? I mean, this is kind of next level stuff, but this is what people are thinking about. So all of the factors that you said are true, and they're the reason that, as Elizabeth said, we can't get all our patients to CAR T. But I'm very hopeful that because all of you are pushing us forward, telling us how much need there is, that there will be these ideas to bring multiple efficiency ideas together. I can't change the supply chain, but there are ways to do non-viral engineering that might help that as well.
Hi, I have a little bit of a 2-part question. My first, and you may have addressed this, is durability of CAR T cell dependent on your genotype at all? Have you noticed that in studies? Is there any correlation? And then the second question is, with CAR T cell therapy, where does vaccine therapy come into play with that? And with respect to durability and progression-free survival?
I can answer. Regarding genotype, do you mean cytogenetics in fish?
Cytogenetics, that's what I'm
Yes.
sorry.
Okay. Thank you. That's a very good question. The high-risk patients with in the CAR T trials have done less well. Of course, better patients do better. Everybody, what that means, we're not doing a good job. We are not doing a good job for high-risk patients. This is one of we all say this, that we're doing a horrible job for these patients. We're doing better. But yes, having 17p, 1420, all these things, they don't do as well. And that's true in both of the CAR T cell therapy trials. And then your second question was about,
durability of CAR T cells
Yeah.
in the long run. And so does vaccine therapy? I know that-
vaccine therapy, yes.
Correct.
Okay, so you mean myeloma vaccine or like,
Correct.
okay.
No, myeloma
Yeah, sorry,
vaccine,
so
I'm
many
sorry.
vaccines going on.
Yes.
Okay, so there's a thought that potentially after you give the CAR T cell therapy, remember I told you have to get chemotherapy, and then that makes room for these cells, and you get the CAR T. There's a thought that afterwards there's a sort of secondary immune regeneration, and at that time you could potentially vaccinate people with their own myeloma proteins or a specific myeloma protein. My colleague, Dr. David Avigan, who's at BI Deaconess in Boston, has been hoping to get this to be a secondary trial after a CAR T cell therapy. So that's a possibility. I think myeloma vaccine, and we participated in the trial as well, and you know about the trial. So far, it hasn't shown that it's better at achieving a deeper response, but it does have changes in immune responses. So what we're hoping is that we can harness this reset of the immune system with CAR T and then give the vaccine and maybe extend the time that the patients use the CAR Ts to kill and then their own immune system to kill the myeloma.
Maybe I'll just add briefly because there are a lot of questions about CAR T cells. So we have currently 2 CAR T cells approved for myeloma. They are both targeting BCMA. They are both autologous. It's the ida cell and the cilta cell products. Where the field is going here, what you hear from the whole panel here is that there are allogeneic CAR T cells, meaning that there could be donors that donate blood cells and they could be banked, could be off the shelf. That sounds like, why aren't we doing that already? So there are a lot of practical hurdles there. So if you give one person's cells to another person, that is really like a transplant. So it could trigger graft versus host disease. So in order to overcome that, you have to do sort of genomic editing. You have to turn off CD52 and you have to give CD52 antibodies. And if you do CD52 antibodies, you turn off certain immune cells. You could now run into infections and things like that. It seems to so far actually work quite well. There is one product that is very far along that is going for FDA review, an allogeneic CAR T cell. Also, there are developments using multiple targets. We touched before on GPRC5D and BCMA. There was a paper in Science Translational Medicine about 2.5 years ago showing that you can have even better responses, seems to have longer durability if you go after 2 targets. There are also attempts using other vehicles. We talk about CAR T cells, but you could do CAR NK cells or CAR macrophages or CAR whatever. So you could use other types of vehicles. And then Nina is talking about that you could do them in vivo. That is basically, that's how the company that became the Pfizer COVID vaccine actually was started. So the idea was to do an anti-cancer vaccine. It was very difficult. And then COVID happened. They said, let's do a COVID vaccine instead. And that became the Pfizer vaccine. Now that company is going back doing cancer development. So I think that those types of technologies probably could turn into CAR T cells that you give a shot of sequence that goes into the body. And if you do it right, that sequence gets inserted in the right cells. And then those cells become the T cells or NKs or macrophages in the body of the person. So the whole field could go basically into vaccine. It's not the type of vaccines that we think of in general terms when we get vaccinated. Usually we get vaccinated with proteins and the immune system reacts. So these are vaccines where you insert genetic programs. It's just the beginning of the journey. Everything is going to change. And then we have all the antibodies. They are already here. So we have one antibody probably approved in August this year, bispecific. There are probably 4 more antibodies that will be approved within 12 more months for myeloma. And if you add 6 more months, there are 3 more antibodies that are waiting. If all this is in the public domain, you can go online and see. If you know where to look, there are 10 antibodies that are moving with the speed of light. So everything is changing, so for the better.
We have a questions. Let me do the online. Oh, actually, you get. Let me do the online, and I'll come over there because we'll do that. This is directed to Dr. Landgren, but it's for the whole group. I agree that using MRD to decide treatment is an unknown. I'm wondering whether watching trending is a better indicator.
So watching trending mean that if it stays stable, negative, or if it stays stable, positive? Yes, of course. And I think that the more you think about like what is really MRD testing, it's basically you push yourself to develop such a sensitive test that you really want to find or make sure that that last cell has been taken away so the patient is cured. But until you can completely say that you have done that, you are maybe not finding the last cells. The test may not be sensitive enough. So basically it's kind of a threshold somewhere between the real cure and on the slope down there. So for people who are negative, there could still be some cells detectable. And if you make the test a little bit less sensitive, you could find cells in those individuals. But I think if you track either detectable cells or you cannot find them, they're under the threshold, it is sort of the same thing. As long as they don't become more, as long as they don't become active, I think it's the same thing. And there are many patients, talking about this earlier, there are many patients that never reach even a complete response. There are many patients that are not MRD negative. 10 years later, they do great and their disease has not become active. I have patients, I have one lady, she has followed me. I diagnosed her in Washington, DC. She followed me to New York. She followed me to Florida. I think she's been followed for almost 15 years. Her M spike is still 0.3. So not every individual needs to reach MRD negativity, but on average, MRD negativity is probably a good thing to achieve, try to achieve. But some people, they never achieve it and they just stay at that level. So it's really, I agree, it's about tracking, no change, staying negative or staying at the same low level. That's really what we are trying to do.
Oh, thank you, doctors. Talking about vaccines, okay, which is going on now. I see some of us are wearing masks, some of us aren't. I was offered randomly, I'm a patient at Mayo Clinic in Rochester, to get the UV shield. But it was given in Tampa and I just couldn't make it to Tampa to get it. What's your take on all these for the myeloma patients and how do we deal with?
So usually we give it to patients that are immunocompromised. And as you know, myeloma patients are usually immunocompromised. So it is actually accessible to all patients. And what it does is it decreases, if you then get COVID, then it decreases how severe the COVID is and also can be protective. So we started giving it in the inpatient service. So to the sickest patients that we had in the hospital initially, and as there has been more access to EvoShield later on, then we've been giving it to the clinic. And we are giving it, we started then giving it mostly to patients that were on CD30 antibodies, because those sometimes can increase your risk of infections, and then patients on chemotherapy, different types of chemotherapy. But nowadays, since there's a lot of accessibility for it, we give it to most of our patients.
I think it's just to add on to that, I think the patients who I really make sure we give it to, there's two populations. One, right after autologous transplant, like out the door, they get Evusheld. And then patients who've had BCMA directed CAR T or on bispecific therapy, because there's such profound decreases in their ability to make antibodies and also for their ability to respond to the vaccine. Many of them don't respond. And so those are the people that I really do make sure they got their Evusheld. People on daratumumab, which many of you may have been on or are on. We probably should be more aggressive than we're being, but we haven't had as bad of an outcome there with COVID, et cetera. So I think that's a little bit of a gray zone. So that's kind of where we fall.
I have a question that may have been touched on partially, but what do you say to patients who getting at or near remission who nevertheless fear that for the rest of their lives, they'll be on the same kind of treatment and or feel that it's ephemeral and not real?
You've been the longest practicing, you answer that.
So the question is, if you reach complete response, do I have to be on therapy forever? Is that the question? Well, so that's a very common question. It's a great question. So the answer is that there is no formal reference book answering what the optimal duration of therapy is. All the trials usually have continued therapy. The clinical guidelines say that it's justifiable to offer continuous therapy. But I've treated patients for many years who have been negative. And I've, as I mentioned, studied MRD testing for a long time. So I have many patients who have told me, I've been MRD negative for a few years now. I'm going to stop the therapy. And I have many patients like that I keep on following. And they just remain MRD negative for the most part. So there are patients that have been MRD negative for a few years in a row that have just decided to go off. There are some individuals who say, I'm MRD negative, I don't have any side effects, I don't want to jinx it. There are some patients who have gone off therapy when the disease years later has come back. But I have, with my experience, this is based on my clinical experience, not found that if the disease has come back after years, that it has been more difficult to treat. So here's my prediction, and this is just my personal observation and thinking. I think that the field is going to go towards blood-based MRD tracking. And I think that we will have situations where patients can be MRD negative for some time point, say 2 or 3 years or something like that. These are round numbers. And there could probably be people deciding to go off therapy. And if you have a blood-based MRD test, that means you don't have to do a bone marrow biopsy every time. I could see the way I think that people could choose to be tested, say, every 3 months with a blood test and really make sure there is no recurring disease. And if you see that many, many times, that person probably could be a potential cured patient. The longest follow-up patient I have that has been off therapy, I have a lot of patients that were treated by Dr. Barlogi, who is no longer practicing. And when he retired, many of them came to me at Sloan Kettering. And now when I moved down here, they are here. I have some patients who have been off therapy over 20 years. And they remain MRD negative. So I think myeloma can be cured, and I'm sure Elizabeth and Nina agree. The problem is we don't know who those patients are up front. But I think with better and better drugs, we can take more patients into MRD negative. And I think if we can confirm that there is sustained MRD negativity, that is probably the path towards that goal. And there are more and more patients reaching that. So I think developing more blood-based tests will be the way to advance the field. And we are trying to set up a lot of those tests, and we have a lot going on here in Miami.
One thing I want to add, first of all, 2 things I want to get rid of for myeloma patients. 1, bone marrow biopsies, and 2, urine protein electrophoresis, the jug. I hate that thing. So I'm actually trying to do a study called Urine Luck to prove that we don't need that anymore, but that's a side thing. But the one place I think it's difficult, and this is what the master trial is showing us. This is a trial looking at very intense therapy, DARA and KRD and transplant, and more DARA and KRD, and it's very effective. But the patients that are not able to get off therapy for a long time are the high-risk patients. So even if you achieve MRD negativity, if you have high-risk disease by DNA, or if your doctor tells you you have high-risk disease, those are the patients I feel very hesitant to stop the therapy on. But I agree with Ola, there are so many patients, they're chilling, and then, they're like, okay, I'll take my risk. I will take my risk, and they've done okay.